Well, now for the rest of the story on DNA replication. I'm going to talk a little bit about where the energy comes from to put these molecules together, as well as the 3' and 5', some of the details about the structure of DNA. I'm talking about the lagging strand. It's always the toughest bit. One of those details. You have the basics, but this is at least a little bit of the rest of the story. One of the things that I think is a good framework for thinking is that this is not an engineered process. You'll hear some of this stuff and you'll watch it and you say, that seems weird. It seems like there's a lot better way of doing that. I agree, but it's not engineered. It's just a biological process, and this is how it happens. It's a process that we discovered, we didn't invent. Let's take a look. Quick review of the basics. Here we have double-stranded DNA. It gets split apart. We put in complementary bases on each, and this was done by helicase, which did the splitting, and DNA polymerase, which laid down the complementary bases and bound them together with covalent bonds. Now you're all up to speed. So in terms of the energy required, this is an expensive process. Remember, it happens just before cell division, which is also an energy expensive process. So anytime we grow things, whether that's growing into an adult, replacing our skin, growing more hair, really energy expensive process. So with DNA replication specifically, helicase takes energy to split those strands apart. And the nucleotides, when they get added and assembled into a larger molecule, that also takes energy. Let's take a closer look at that here. Well, here we can see just the basic cartoon version of DNA being synthesized, and over here we're seeing more detail. Now here, the sugars and phosphates have been bound together with covalent bonds, and this new one is coming in. And this is adenosine, and it carries these 3 phosphates, just like our friend ATP. So this A, adenosine, is the same as this adenine over here that makes up DNA. And this is a really common molecule. We'll see it all throughout the course. It has 3 phosphates, so that stands for adenosine tri, or 3 phosphates. And here we see those 3 phosphates over here. When any of these bonds are broken, they release energy, and that allows us to do work. And so that's what's happening over here, is these get broken, release energy, and that allows a bond to be formed with the other nucleotide. And this is true not only for adenine, but for the rest of them as well. So all of our nucleotides, A, G, C, and T, have a 3-phosphate version, gives them the energy they need to bond. Well, the first problem in DNA replication that may seem a little strange is just how to begin. Where do you begin? So you're DNA polymerase, and you just can't seem to get a handle on this. So here I've drawn a prokaryotic or bacterial bit of DNA. Their chromosome is just a single chromosome in a big loop. But even then, DNA polymerase just can't seem to get in there. Even if helicase comes in and separates the DNA strands, it just can't get a hold and get started. And so how to begin is a real problem. What it needs is a primer, a little bit of RNA in there, and that's laid down by a different enzyme known as primase. Then DNA polymerase can come in and bind to this and start tearing down the tracks and laying down new DNA. But getting started is a bit of a trick, even more so in eukaryotic organisms. Take a look at this one. So same sort of setup here. Polymerase is just confused at this point in terms of where to start until primase comes in, lays down a primer. Now DNA polymerase is less confused and can go over and bind there. The other thing I want to bring up here is that normally these long chromosomes are started off with a primer maybe at the end, but there's another one that starts here and here and here, so that DNA replication can continue it's several points all at once rather than going from one end to the other, which speeds up DNA replication a whole lot. So in eukaryotic organisms with these linear chromosomes, the other problem that happens is you have this primer left on this end and on this end that were required to get polymerase started. So primates did a good job. This got started, made the rest of the DNA, and now I have my sister chromatids all made and ready to go. But what do I do with this? Well, I can kick it off, but DNA polymerase can't come in and fill in the rest of this. So it just gets deleted. And same with this one over here. They just keep getting whittled away. And so each time the DNA replicates, a little more of the ends of those chromosomes keep getting eaten off. I know, it's frightening. What are we to do? Well, all the good stuff with the genes that help us make proteins is in here in the middle. But on either end of all of our chromosomes, we have telomeres. These bits of DNA, they're just repetitive segments that don't code for anything. They do absolutely nothing other than just sit there and wait to get deleted and protect the good stuff on the inside. It's a pretty interesting workaround, but it keeps us safe. And the next problem is the gaps. This is actually not that hard to solve, but here's why it's a problem. So here again we have that prokaryotic chromosome, single ring of DNA. We have the primer here, laid down this orange strip of DNA all around it, got to the end. Even if we kick this primer out, there's still not a way that polymerase has of binding this strand of DNA to the other end. Fortunately, there's only one of these gaps in prokaryotes. There's more of them in eukaryotes, but let's take a look at their solution. On a long strand of DNA like this, there might be lots of gaps. And so here we're just looking at a small segment of the DNA with those sugar phosphate backbones here and one down here. Oh, there's a gap. There's a covalent bond I didn't make. And so this strand over here is actually not bound to this one. That could cause some real problems. So the solution is another enzyme, DNA ligase. It swoops in, makes the bond, saves the day. Problem solved. So our final problem is the other strand. What do you mean the other strand? You know, the other strand. So this whole time we've been talking about this strand right here, and DNA polymerase moves from the 3' end to the 5' end. Well, wait a minute. What do you mean by the 3' and 5'? Let's take a look at the chemistry behind that term. Well, let's go back to our basic nucleotide layout. There's several carbons, they weren't drawn in. All right, well, there's this shortcut that we use where we don't draw in carbons, but they're there at every corner. So I'm going to draw in those carbons so we can see them. And there they are. This is a 5-carbon sugar. This is deoxyribose, this guy right here. And so we start labeling them just so we can talk about them. There's the 1', 2', but the key ones are 3' and the 5' carbons. And the 5' carbon is the one bound to the phosphate that gets bound to the next nucleotide. Let's look at a string of them so we can get our sense of it. So let's go down here. This is the 3' end down here. So this 3' carbon right here, next to this oxygen and hydrogen, that is that carbon right there. And here's the 4' carbon and the 5' carbon bound to the phosphate, bound to the next 3 carbon, 4 and 5, bound to another phosphate, 3, 4, 5, and it ends right here with the 5' carbon and the phosphate. So you can kind of recognize the 5' end, which is the one with the phosphate sticking out. And the other end goes the opposite way. 3' carbon is on that side, and it runs the opposite direction toward the 5' end with the phosphate sticking out on this end. Well, there's the chemistry, but I kind of like to think of it like a road. Both roads in DNA, we say, are arranged in anti-parallel fashion. So they're parallel to each other. You can tell these strands are going in a parallel direction, and you can definitely see it when we simplify it here. And it's just like a two-lane road, right? These lanes are parallel. This car's going to the right and that car's going to the left. And as long as they stay in the correct lanes, nobody crashes. And DNA is kind of similar. DNA polymerase can move along this direction on that strand, and everything works just fine because that's how DNA polymerase is programmed. It moves from 3' to 5'. I tend to think of these as just directional terms. Don't worry as much about the chemistry. Think of them like right or left, north and south, 3', 5'. They're just directional terms. And that's the way that DNA polymerase has to go. Well, the problem is that it wants to move this direction on both strands at once. But how can it go forwards and backwards at the same time? Because this strand is arranged backwards and it can't go 3' to 5' by moving this way. But it can, because this is a chalkboard and you may be looking at it on paper or a screen. Those are all flat. DNA is not flat. It's a double helix, and furthermore, it is a flexible molecule. It's like a long thread. Let me look at it, have you look at it this way. So here's our two strands, right? We got 3' and 5'. And if I had a molecule moving along this way, how could I go 3' to 5' on that one? What if we just turn this one around? And now we're going 3' to 5' on both sides. And that's kind of what DNA does. It's more of a nice trick. It's not like you're going to have to flip your paper around and bend it to do this, so don't worry about that too much. But it is possible, and sometimes we just need to kind of free our mind from the constraints of the page to understand it. Let's take a closer look on the screen to get an idea about why this is a problem and how these molecular complexes of enzymes work together. Here's a strand of DNA undergoing replication. Let me draw your attention first to some characters that we already know. DNA polymerase. There's another one down here. There are actually different versions of them, which we're not going to concern ourselves with too much. Here's ligase, closing up those gaps. Here's primase, laying down RNA primers. And helicase, along with another enzyme that helps with the twisting motion called topoisomerase. So they're splitting apart the DNA strands. Now, we have two different strands. The strand we've been talking about so far has really been the leading strand. We started down on this end, reading at the 3' end and synthesizing with DNA polymerase in the direction of 5' to 3', continuously, no problem. It just keeps chugging, and we're not really worried about that one. That's kind of the model we've been using so far. This other strand, we call the lagging strand, it's the problem one. It's the other strand. It's the one that we had to flip around this way so that it was facing the right direction. Now that's a little too difficult to conceptualize at the same time as we talk about it, so we'll look at it this way. So we can treat this lagging strand in 3 steps. First the primase, second polymerase, and third ligase, because that's the order things happen in. So first, primase lays down the primer, which allows polymerase to come in and do its job and lay down this next fragment until it gets to this segment where the old primer was and there's a gap, which is where ligase, you can see over here, is sealing that up. Now this is done in fragments, or Okazaki fragments, of several hundred nucleotides each, and this way the entire complex of enzymes can keep moving down the DNA strand in that direction, looping out this lagging strand in little bits to be synthesized one piece at a time. A lot more complicated than the leading strand for sure. Well, there's the rest of the story on DNA replication. Blows the mind, doesn't it? There's so many fascinating details in there. But what are you supposed to take home from this? So one thing I would say is when scientists discover things that are amazing and quirky and really cool, they like to share them. Two, there are some practical application, like telomeres. They have a role in aging. It seems that the shorter telomeres get, the sort of more worn out cells become. So that research may contribute to prolonging life and preventing premature aging. So there's an upside to one of them. And third, the way cells work is just kind of quirky, right? There are these sort of problems caused by a process that there's a bizarre workaround for. This is not an engineered machine. The cell is a living organism full of complexity and really sort of innovative solutions, just like us.